Thank you very much, Daniel. Good afternoon, or from Austria, I shall say good morning, everyone. In Austria, it's 9 o'clock a.m. now. It's my great pleasure to be your guest speaker this afternoon, Korean afternoon, and give you an overview of TissueGnostics as a company, but in particular about our TissueFax technology platform, as well as the applications that can be done with it. I will start with the basics, how it works, then show a number of different applications that have been done so far by the global research community using this instrumentation, and finally make our way to the high art of spatial immunophenotyping. I'm one of the founders of TissueGnostics and also inventors of the technology, and currently the Chief Executive Officer of TissueGnostics Global. And as a side branch of my career, I'm also a visiting fellow at Queensland University of Technology in Brisbane, Australia, where I work on artificial intelligence. To start with, I would like to introduce you to a survey that has been published a few years ago in Nature. A survey that was made among 1,500 scientists. And there was a questionnaire, and the outcome of this survey was that more than 70% of researchers have problems in reproducing published data. And another 20% even have problems in reproducing their own data. So the question has been raised, is there a reproducibility crisis in science? And a call has also been raised for three things. First, the better mentorship. This is something universities and academic institutions have to take care of. The proper use of statistics. This is something each individual PI or laboratory has to take care of. And third, a more robust experimental design, more standardization, observer independence, and reproducibility. This is something where TissueGnostics, as a medtech company and provider of high-end analytical instruments, comes into play. There are a number of technologies available which provide observer independence and standardization for measurement of cellular parameters and markers. One that everyone knows, obviously, is flow cytometry. This technology has been available for decades now. And if you look in public Medline and search for the term flow cytometry or FACS, F-A-C-S, fluorescence activated cell sorter, you will literally find thousands of papers that have been published over the last 50 years using this technology. Flow cytometry offers a phenotypic and functional analysis. However, a major limitation of flow cytometry is, that's what the name flow cytometry suggests, that it can only be applied to cells in suspension, like blood cell or cultured cells. So, as we all know, the majority of the human body consists of solid tissue, not of liquid cells or liquids. So it's not appropriate to analyze lung, liver, kidney, prostate in a flow cytometer. Therefore, we have developed our TissueFax cytometry platform, the microscopic equivalent to flow cytometry, which also offers a phenotypic and functional analysis, but now on slides. And that means in images. Slides get scanned and image analysis, image cytometry is performed. So what we refer to as TissueFax cytometry, that is in fact image cytometry, the specific way TissueGnostics is doing it. A few words about my company. I already said I'm one of the inventors of the technologies and also co-owner and CEO. The company was founded in April 2003. So this year we celebrated our 18th anniversary. The headquarters is in this nice building in the city of Vienna in Austria in the European Union. And we have a number of international subsidiaries and branch offices in Europe, in the United States, in Asia Pacific, in Africa, and Brazil. At the moment, our installation base is in more than 60 countries on all continents except Antarctica, and we are approaching 1,800 reference publications from the clients around the world. TissueGnostics, from its very beginning, has always been closely related with research, also collaborative research with academia. A few projects that are currently ongoing, I would like to introduce you. One project is called Deep NUC, Deep Learning for Improved Nuclear Segmentation and Knowledge Transfer Methods in Microscopic Images. This is a collaborative project with Medical University of Vienna, more specifically with the Institute for Pathophysiology and the Center for Medical Statistics, Informatics and Intelligent Systems. Here we are joining forces to develop new solutions for highest accuracy of automation using deep learning technologies. As well, in the same direction is our industry partnership in the S3AI project, which is a COMET Center of Excellence in Austria, headed by the Software Competence Center Hagen. This deals with security and safety for shared artificial intelligence by deep model design, where we aim at the creation of meta-classifiers in this stage for prostate cancer that learn from existing technology. The basic idea is, or the basic challenge is at the moment with machine learning, that you always start from the beginning. You always means at the image you have to teach the algorithm what is a prostate cancer. And when you move to, say, bladder, you again start at the beginning. While human beings, doctors, for example, when they work on prostate cancer, when they have been educated on prostate cancer and then move to bladder cancer, they go back to kindergarten and make their entire education again until they can diagnose a bladder cancer. They directly learn from their knowledge obtained in prostate cancer and transfer it to bladder cancer with the necessary adjustments. While computers still today go kind of back to kindergarten and start all over from the very beginning again. And this project focuses on transfer learning. How can computers learn from existing models in order to avoid that we always start at the very beginning when we train a machine to recognize something like different types of cancers. And this is, in fact, a project that we have initiated at Queensland University of Technology. Here we are working on an AI-based decision support system for clinical pathology, which we call the virtual histopathologist for precision cancer diagnostics. And we have, I think, impressive number of international academic partners from all over the world, as you can see here. TissueGnostics has also been involved in 5 high-profile EU-funded Marie Curie innovative training networks. They were focused on different topics like Alcatraz, translational oncology on the ALK-related malignancy. ALK is the abbreviation for anaplastic lymphoma kinase. Then 8-path, this was headed by University of Cambridge in UK. 8-path digital pathology, which was headed by University of Nottingham and University in Spain. Then the calcium sensing receptor biomedicine. The calcium sensing receptor is a potential target for therapeutic intervention in a number of non-communicable diseases like breast cancer, diabetes, asthma, or Alzheimer. And INSEM, this project was headed by Medical University of Vienna. INSEM from Technical University of Aachen in Germany, the European Network on Cell Migration Studies, was focusing on quantifying migration of cells in tissue. And those 4 projects are already finished. What is currently ongoing is Helical, headed by Trinity College in Dublin, which focuses on big data analysis for clinical benefit. For TissueGnostics, the common denominator in all these EU projects, each of those was funded with 3 to 5 million euro. The common denominator for TissueGnostics is always high-end, state-of-the-art solutions in image cytometry for quantifying molecular markers in tissue sections. In order to obtain this goal, TissueGnostics has developed and launched a set of products, a range of products, which start with this budget solution of a bright field scanner for 2 slides, which is named StrataFax 2, to our TissueFax family of products, which are also available in different configuration, like TissueFax Histo, which is for bright field scanning only, TissueFax Flu for fluorescence, or the combination of which is TissueFax Plus. As you can see at the hardware, at the core of the hardware is a high-end microscope of Carl Zeiss. So TissueGnostics is an OEM partner of Carl Zeiss Germany. We have a scanning stage for 8 slides. All objective lenses that Carl Zeiss offers can be used from 2.5x to 100x, including oil immersion or water immersion lenses. We have an automated oiling system, for example, and we scan also slides on or well plates on inverted system. This is then called TissueFax I like inverted, where you can do also live cell imaging or time kinetic assays. This can be combined with a culture chamber, then you can really do live cell imaging. For high throughput scanning, we have TissueFax SL with a slide loader for batch mode scanning of up to 120 slides. And each fluorescent configuration of TissueFax can be equipped with a spinning disc confocal for whole slide confocal imaging. So this is the range of products that we offer to the global life science community. The immediate output of all of those microscope-based scanning systems is, like with any other scanner that is available on the market, a virtual slide. Here we see an example of an entire mouse leg. This we received from a group at University of Sheffield in UK. The virtual slide consists of many, maybe even thousands of individual fields of view, which are scanned with overlap and pixel-wise matched. So you do not need to take care of individual fields of view, but you work with your virtual slide and the software does the rest. You can zoom in to see more and more details, even cellular and subcellular details. The same obviously works in fluorescence. Here we see 4-color immunofluorescence of prostate. These are the individual prostatic glands. In blue is DAPI, the nuclear marker. In red is cytokeratin as a secretory epithelial cells. In green here are the basal epithelial cells. And in orange is Amaterasu, AMACR, and tumor marker. And we can really see here how the tumor invades the normal tissue. This is like a fight scene that we see here. The tumor invades the healthy tissue and converts prostate into a malignant transformation, malignantly transformed tissue. Here we see a prostatic stone, and we can also zoom in to see more and more details down to the subcellular details, where we have here the individual nuclei and the cytoplasm of the individual secretory cells. This is the lumen of the gland, the interstitium in the tissue. Depending on which system configuration you're working with, you can acquire up to 10 fluorescent channels per field of view, so in case of fluorescence acquisition. Now, as I said, at the heart of a TissueFax is a Zeiss microscope. But still, I would like to make very clear that TissueFax is more than a Zeiss microscope with software. It is a tissue cytometer. TissueFax cytometry is based on cutting-edge segmentation algorithms, for single cells and histological entities in tissue sections, but also in cell cultures. And these segmentation algorithms are either based on high-end image analysis, but also on deep learning algorithms. So what is the difference between a microscope with software, which you can buy anywhere with any supplier, and the tissue flux cytometer? First, it is the degree of automation and the workflow design that we have created. Second, it is the quality of image analysis. First, you have the highest quality from the microscope part. Carl Zeiss is one of the leading suppliers of microscopy solutions, but also in combination with TissueGnostics, the high-end algorithm, which give you individual cells in tissue section. And finally, it's this flow cytometry-like handling of data, of an interactive working with your single cells in tissue sections, which we will see in the slides to come. There are three image analysis or image cytometry solutions that we offer. They're called HistoQuest for histological samples, so bright field images only, single cell recognition in immunohistochemistry, TissueQuest, the single cell recognition in fluorescent samples, and StrataQuest, the high-end solution for contextual tissue cytometry. We can say that StrataQuest can do everything which TissueQuest and HistoQuest can do, and much more, including it has a classifier for automatic detection of histological entities like glands or vessels or tumor areas, and also deep learning algorithms. Now, let's look on how those applications can be achieved by high-end imaging. As I said, each fluorescent combination can be equipped with a whole slide confocal spinning disk device. We see here from one of our clients, Janelia Farm Research Campus in Ashburn, Virginia, a mouse brain section that has been scanned. In order to acquire the entire mouse brain, 350 individual fields of view had to be acquired. So the tiny rectangles that you see here are in fact not tiny. These are the individual high power fields of view. Each field of view consists of 4 channels, and each channel consists of 21 layers. So in total, 29,400 images have been acquired, which are 115 gigabytes of compressed data. And that took a 20x scanning 1.5 hours. If you only scan in one focal plane, it's done in 8.5 minutes. But also 1.5 hours is very fast. If you try to acquire 29,400 images manually, you will sit several weeks, and overall it will not be possible. You will fail. And now we can zoom in, like in Google Earth, we can navigate through the sample and zoom in to see the cellular and even subcellular details. The system, all the systems of TissueGnostics have IVD compliance, so can be used for in vitro diagnostics applications. The company as such has been certified for ISO 13385, the main quality norm for medical products, first time in 2008. And we are currently going through a recertification process according to the new ISO 13485 standard from 2016 and the European Medical Device Regulation, which will become effective next year in May 2022. There is a nice workflow to analyze immunohistochemically stained consecutive sections. So when you have a tissue block and cut several slices of the salami and stain them with different markers, you can, for example, identify the tumor area on the H&E slide and then analyze this area on slides which are stained with different markers. So the basis of this is a process called image registration, so that the computer aligns consecutive images in a way that it knows which part in section 1, which area in section 1 relates to the same area in section 2, section 3, section 4, and so on. Obviously, you cannot do a co-expression analysis on a single cell level, because through to the physical sectioning process in section 3, 4, and 5, different different cell layers will be visible than in section 1, but you can compare regions like tumor areas versus normal adjacent tissue. Also, trichrome staining can be analyzed, and you can automatically identify, for example, big glands with lumens, small glands without lumens, blood vessels, and stroma. Here is another slide for what I have shown earlier, this early identification of malignant transformation in prostate cancer by using 4 markers. Here we have the normal area. In normal prostate, the healthy ducts come with this basal epithelial cell layer. After malignant transformation, this layer gets lost, and the secretory epithelial cells express this marker, AMACR, a marker, and the combination of both the loss of the basal epithelial cell layer and the expression of the AMAC is a sign for malignant transformation, which can be quantified here. So you see here in the normal area, you have a population of basal epithelial cell marker positive cells at the glans, but no significant expression of the AMAC, while on tumor glands, the basal epithelial cell layer gets lost. This quadrant is empty, and you have a clear positive, in this case, 35% tumor marker positive cells. So rather than visually looking at it and counting with your finger on the monitor, you get a single cell-based quantification in an observer-independent manner. In order to identify single cells and quantify the marker on single cells, we use the nuclear stain typically as what we call the master channel. This applies to both immunohistochemistry as well as immunofluorescence. In this example, we see a colon section with a double stain. In blue is what is called the counter stain, hematoxylin, that labels all the nuclei. And in brown is Ki67, a proliferation marker. So the obvious question is, how many cells are proliferating, or in other words, how many of the blue nuclei are also stained in brown? We made internally also a survey among approximately 200 scientists and asked them what they think, how many cells are positive in this sample in percentage. And the surprising answers that we got ranged from 1 to 40%. So, and let me point out, these were estimations from experts, not from ordinary people on the street, but all from researchers in the field. Still, we get such a wide range of estimations from 1 to 40%, which shows the main problem associated with visual evaluation. We always get a range of estimations rather than a reproducible value, a measurement. This may even depend on the light conditions around, or if the researcher or the person who is doing the analysis is tired, or does it in the morning, or is stressed, or has a lot of time and can thoroughly work on it. So this is the problem of visual evaluation. When we analyze this sample with our TissueFox platform, we use, as I said, the nuclear marker as a master channel. Here you see in yellow what the algorithm has found as individual nuclei, as individual cells. And then we measure for each nucleus how much blue and how much brown is there. And this is shown in such a scattergram. People in flow cytometry would call it a dot plot, just that we see here on the x-axis now hematoxylin, and on the y-axis the brown marker, in this case our Ki67. So there is this important line, well known from flow cytometry. It's called the cutoff. The cutoff has to be determined using an isotype matched negative control, and it defines the specificity of our staining. Only cells with a brown intensity greater than 20, so this population, will be considered as positive. What is below the cutoff will be considered as negative. And here we see we have 14.5% of positive cells. And The important thing is we will always get the same result, whether we do the analysis on Monday morning, on Wednesday noon, on Friday afternoon, if we do it in Austria, in Korea, in Australia, in the US, we will get a reproducible measurement rather than a range of estimations. This was the simple case when the Ki67 is expressed in the nucleus, when our marker is expressed inside the nucleus. Many molecules that we are looking at are also expressed in the cytoplasm or are only expressed in the cytoplasm, but not in the nucleus. Then we need a modification of the approach. We still use the nucleus as master channel, but then we generate a secondary measure mask around the nucleus, which covers the cytoplasm. And here we see such an example of a skin. This is from skin cancer research. The marker is called COP1, but this doesn't matter. It's about the methodology of detection. You see that the marker is constitutively expressed, but the nuclei are negative. So the black holes we see here are the nuclei, and the marker expression is around the nucleus in the cytoplasm. By using the nucleus as a master channel and then measuring around the cytoplasm, we can quantify this cytoplasmic markers like this as well. We have here on the left-hand side a normal skin, and in the center a biopsy taken after UV irradiation. And on the right-hand side is the negative control, the isotype matched negative control. So normally people would look at this and say, okay, this is 1+ positive and this is 2+ positive. And then maybe your colleague comes and says, well, I would say this is 3+ positive. Again, observer biased estimation. Instead of that, our TissueFox platform will give you an observer independent measurement. Here we see the normal skin, and on the right-hand side, after UV irradiation. What we see at a glance is that the number of positive cells increases from 16.8 to 44.7%. But what we also see here is the shift of the population towards higher values. In the normal skin, the highest values are here around 90. But after UV irradiation, this goes up to 130. So what we visually perceive as brighter in this image is shown by this shift towards higher values. And this is, again, an observer-independent measurement on the single-cell level directly in the tissue, so an in situ analysis. So now we have seen that we can quantify the nucleus and the cytoplasm. We can also look on subcellular structures or cellular compartments inside the cells. We have special algorithms to quantify FISH signals, for example, or other globular staining pattern, or the RNA in situ hybridization, including RNA scope, where you assign hundreds or even thousands of dots to the individual nuclei around which they are positioned. So again, you get a single cell-based analysis of these genetic markers. But I would also like to briefly mention that other structures can be identified. I don't have the time to show everything, but it could be like neuronal tracing or actin filaments or vimentin, so cytoskeletal components, but there's published work about it. But what I would like to show you is a very interesting application which we developed with the Department of Immunology in Regensburg in Germany, in Professor Uwe Ritter's lab, and this is the quantification of intracellular protozoan parasites. We're using here leishmaniasis as a model. These are the parasites which are transmitted through mosquitoes, and they infect specific cells like macrophages in the body, and patients suffering from this disease really see substantial tissue damage. So I'm not going to talk about the disease a lot. I'm focusing on the analysis. In Regensburg, they're working with an inverted system of TissueFox where they can use macrophage cultures in micro well plates. Here we see one well. The individual fields of view that had to be acquired at 20x in order to cover the entire well. And when we zoom in here, we see the individual nuclei. The big blue spots are the nuclei of the macrophages, and the tiny blue spots are the nuclei of the parasites. We first identify the nuclei of the parasites, and then make this what is called ring mask, a ring around each nucleus, and measure the dots inside. The algorithm, of course, is a mathematical procedure. An algorithm does not understand what is a parasite, right? It deals with pixels and intensities and pattern in the image. So we have to provide controls in order to set and define the sensitivity of our algorithm. Here in the lower panel, we see control, which does not contain any parasites. So what we see here in this image would all be false positive. So we use this control in order to set the sensitivity of the algorithm in a way that it does not find anything, and then apply this setting to our actual test samples. And what we find now here, this is the positive signal. These are parasites. In this publication, there was also a comparison between manual counting on a defined population of cells and automated analysis showing a very good correlation. So this was an example of analyzing subcellular, or in this case, intracellular structures on the single cell level in tissue. One of the features that our clients around the world appreciate most is the ability to track back and forth individual cells from the virtual slide to data space and back. So you see here a part of the virtual slide, and the cells in green are T lymphocytes, in red are monocytes. And when you click on any identified cell in the image, it starts blinking in the scattergrams. So you know exactly where this cell is located in data space. Following our motto, from image to data, we consider this a step forward and call it the forward connection. It also works the other way around, from data space back to the image. Like in flow cytometry, you can create a gate, a selection of a subpopulation of cells, which will then be highlighted in a specific color in the other scattergrams, but also highlighted in the virtual slide. So you see where a certain phenotype of cells is located in your tissue in relation to histology or to morphology. Where in the tissue does a certain cell population, a certain phenotype occur? And so we are moving now towards this spatial immunophenotyping and analysis of bigger structures. A very much appreciated feature is that we can also view or visualize fluorescent images as if they were immunohistochemically stained samples, which is called the Pseudo Ihc View. What we see here is a 5-color immunofluorescence, and we can convert this just for means of display or for visualization into a bright field image. And especially people working in histopathology are very much used to this view and prefer it over the fluorescence view. And although this is stubby, it looks like hematoxylin. And then we can add the other channels. Alexa 488, looking like a chromogen. Cy5, which is in this case a lymphocyte marker. Then we have here in blue the actual epithelial cells. And the fifth, there are only 2 cells. You see here a yellow and here yellow of very few cells which are yellow positive. But we have now 5 different fluorescence markers shown like as if there were an immunofluorescent image. This is the original image, the fluorescent 5-color immunofluorescence, but it's shown as a bright field in Pseudo Ihc view, as a bright field sample. Now, we have seen that we can, with the TissueFox platform, we can scan bright field images, fluorescence images, confocal images. We can identify the nuclei of each cell in the tissue section, the cytoplasm, subcellular structures. This enables us now to address a very important question in medicine, and this is the immune status in situ. It's also called the phenotypic characterization of tissue-infiltrating leukocytes. In flow cytometry, we can determine the immune status systemically in blood. With TissueFax, we can do this for a specific organ, for a specific tissue. Here we see a 5-color staining of colorectal cancer. In dark blue, we have DAPI, the nuclear marker. In green is CD4 T helper cells. In red is CD8 cytotoxic T cells. And in green is FOXP3. You may know that the co-expression of yellow and green, so CD4, FOXP3, characterizes what is called regulatory T cells. So we use the nuclear signal, the DAPI, as a master channel in order to identify the individual nuclei and then measure for each nucleus the immediate surrounding, how much yellow, how much red, and how much green is there. And this is shown in this type of scattergrams, where we see here the CD4 positive population or the CD8 positive population, or ultimately the CD4 FOXP3 double positive population. Like in flow cytometry, the two cutoffs here separate our diagram into 4 quadrants: the upper left, the upper right, the lower left, and the lower right. And depending on where a cell, a dot, but the dot represents a cell, where a dot is positioned, we know which markers these cells express. Here in the upper left, we have the double positive, the co-expression of CD4 FOXP3 in this population. This can also be done in this case of bright field. Here we see an example of benign prostatic hyperplasia done on a biopsy, and brown here is CD45, or on another section CD3, and the scattergrams here show the CD45 staining, or here on the right-hand side, the CD3 staining, and how many cells are present. And this, we analyze the whole biopsy, also measure the area, the size of the biopsy, and can this then normalize to the area. So here we have 227 CD45 positive cells per square millimeter of tissue, and 172 are CD3 positive per square millimeter of tissue. This way you can combine tissue either from different patients, or like in this case, from the same patient in different years. So from 2001, 2003, 2004, then you can check how a disease develops, or in this case, how the immune response against the disease develops. Although in BPH, the immune response is associated with the generation of the disease because there is an autoimmune component. So in TissueGnostics is working in the scope of precision medicine, where we aim to quantify and more markers on the same sample. So not on consecutive sections, but on the same section, so that we can really do a co-expression analysis on the single cell level, not only on the same area of the tissue, but really on the same cells. One way to achieve this and go beyond the traditional limits of fluorescence microscopy, which is maybe 6 markers, the limit occurs due to the spectral overlap of the fluorochromes. One approach to do this is multiplexing. Multiplexing is a technology where you take the tissue sample, stain it with, say, 4 or 5 fluorochromes, and one is DAPI, the nuclear marker, and then 4 antibodies with fluorochromes like Cy2, Cy3, Cy5, and Cy7. You image the sample, and then you inactivate the staining or remove it, and stain. There are different protocols how this can be done. Then you stain the same sample again with the same fluorochromes, but different antibodies. Yeah? And you repeat this a few times and then combine the data set to a virtual 10, 12, 15, 20 color immunofluorescence staining. Yeah? By successive imaging of always 4 or 5 channels in each round. In this publication from Yale University, the workflow, the technology is described. It's based on imaging with the TissueFAC system and a subsequent in silico combination of this virtual multi-channel image using our StrataQuest software. So I don't have the time to go into details, but this is anyhow a published paper, so you can always go into it from Blenman and Bosenberg in the Cytometry. What you end up with is a multicolor single cell analysis, which looks like one of these fantastic flow cytometry data sets, but this is not flow cytometry, it is tissue cytometry. Each dot that you see here represents a single cell in a tissue section. And another approach to increasing the number of markers on the same sample is multispectral imaging. So what we saw a minute ago, multiplexing uses standard microscopy configuration, bandpass filters, for rescanning the same sample multiple times. In multispectral imaging, we use a special hardware, and that is a liquid crystal tunable filter, which limits the detection to a small band of 30 nanometer, but this detection band can be moved in 10 nanometer steps. So what we generate is a Lambda Stack. I assume you're familiar with the term Z-stack or C-stack from confocal microscopes, where we analyze the same field of view in multiple layers and at multiple focus positions. Here we look on the same field of view in the same position, focus position, but acquire in different spectral intervals. So each fluorochrom is acquired several times. And then by using reference spectra of the individual fluorochroms, it can be computed how much of the total emission comes from fluorochrom 1, 2, 3, 4, and so on. So this way you can extract the information from this raw data set, which is very confusing, overexpressed, spectrally overlapping, and generate a highly specific, as we can see here on the right-hand side, unmixed, spectrally unmixed sample. A practical example of this, also with relation to the current situation of our world, has been published last October in Cell. A group at MIT in Boston used TissueFAC spectra in order to quantify marker expression and understand the immune response against the SARS-CoV-2 virus in COVID-19, in the lung of COVID-19 patients. I don't have time to go into details, but they acquired 7 markers, which were spectrally unmixed using this liquid crystal filter technology. They, of course, apply different immunological protocols to the lung tissue of corona patients and found that we have a problem, we all know this nowadays, with long-term immunity. So the concept which some countries, and at the beginning of the pandemic in 2020, still focused on and thought herd immunity will come, this does not work because the SARS-CoV-2 virus tricks the immune system and avoids the formation of what is called germinal centers, which are essential for building long-term immunity. But look into this if you, I don't have the time to discuss this in more detail. This would be a separate talk, in fact, but it's anyhow published. Now, we have seen that we can acquire bright field images, fluorescence images, confocal images, multiplexing technology, multiplexing-based images, multispectral images. We have seen we can analyze single cells, the nucleus, the cytoplasm, subcellular compartments. Now let's focus on the other direction, multicellular structures, histological structures. And for this, we have solutions for machine learning, deep learning, in particular an algorithm for recognition of single cells in dense lymphatic tissue. Here, especially in lymphatic tissue, the cellular density becomes very, very high. The cells are very densely packed, and this is a challenge for computers also for the human eye, by the way, to recognize what is a single cell. And with 2 years ago, we have launched, actually 1 year ago, we have introduced a new algorithm based on deep learning for single cell recognition, which has an unprecedented accuracy in finding individual cells even under dense tissue condition. A machine learning solution is able in our StrataQuest software to find histological entities like glands or here epidermis or different structures in the skin. Yeah? There's no number, there are no limitation to the number of classes that are identified. Here we have 7 different classes which were recognized automatically after a short learning phase. You highlight this is epidermis, this is dermis, this is subcutis, this is fat tissue. After a learning phase of a few minutes, you apply this to the entire sample and get a result like this. Now we can combine all those technologies from multicellular detection to subcellular detection in different microscopic methods to do what we call contextual image cytometry. Here we have a colorectal cancer. This is from a client in also University of Western Australia in Perth with a 5-color staining. Dark blue is DAPI, light blue is the tumor area, cytokeratin. In red is CD4, and in green is FOXP3, and white is Ki67. The question is, how many CD4 positive cells infiltrate the tumor? How does the tumor microenvironment look like? And how many of the CD4 positive cells are FOXP3 positive, so are regulatory T cells? We identify the individual cells, as this shown before, using the nucleus as a master. Then we identify the tumor area using the information from the cytokeratin staining. Here we see in orange the mask, what has been recognized as tumor. And then we can combine these masks and ask a rather complex question. In this case, we ask the question, how many CD4 positive cells are inside the tumor? And if they are outside the tumor, where are they? In this scattergram, we only see CD4 positive cells. So all cells have been identified, but in this scattergram, we have what is called an input gate on CD4. So each dot that you see here represents a CD4 positive cell. And on the y-axis is the distance in micrometer. So the further up, the further away the cell is from the next tumor area. And we can then make gates which show the cells from within 25 to 50, 50 to 100, 100 to 150 micrometer, and so on. And here in the quadrant statistic, we see the detailed analysis, the total count, the relative percentage, and also the cells per square millimeter of tissue. We see the further we go away from the tumor, the less CD4 positive cells are there. We can visualize this. Here we see in orange the tumor area, and in red those CD4 positive lymphocytes which are just outside the tumor, but not further away than 25 micrometer. Or here, those lymphocytes, CD4 positive lymphocytes, which are 25 to 50 micrometer away. So we can do a very detailed analysis on the single cell basis. It's not, estimation and that we say, most of the cells are somewhere, or the infiltration rate is 1+, 2+, 3+ positive. No, you get an exact measurement on the single cell basis in relation to the position of the tissue. How many cells are where in the tissue? What is their phenotype? And this is finally what we call spatial phenotyping. In this case, it's of the tumor microenvironment, but obviously it can also be applied to any other tissue, like, for example, autoimmune disorders. There have been a number of clinical studies by pharma companies like Novartis or Merck on skin diseases like atopic dermatitis or psoriasis, where the same approach has been used. Here we have a 7-channel multispectral imaging. This is the raw data set. And also it's a colon cancer. After unmixing, we get here a very clear, if it was not tumor, I would even say beautiful image, but it's a very specific spectrally unmixed image. These are the individual channels. that you can see. You know, this is one of the immune checkpoint marker stainings. And here you see the quantification. For each cell, you have the dot. So if I double click this cell and I see it blinking in all the scattergrams and know exactly where this cell is located, then we can also make this distance zones and measure the distance to the tumor area, like we have seen in the previous example, and then quantify the individual markers. You have here the CD68. We always use the backward connection and see in red the contours which are to the current gate. In this case, it's CD68. Here is CD8, cytotoxic T lymphocytes, CD4, T helper cells, or in this case, CD4, PD1 double positive cells. And finally, I would like to show you how you can work with these gates to do what is called a gate arithmetic. On the first scattergram, we see PDL1 versus area versus mean intensity. So we define this gate in green, which are our PDL1 positive cells. This gate is used as an input gate in the second scattergram, where we have CD4 versus PD1. We use the lower left quadrant, so the double negative, as input gate in the third scattergram, where we have CD8 versus CD68. And then again, look on the lower left, the double negative. And here you see then the result. And these cells, the red contours here, represent those cells which are PDL1 positive, CD4 negative, PD1 negative, CD8 negative, and CD68 negative. So you can do a single cell, a phenotypic characterization of 7 markers on the single cell level in situ in relation to histology. So to the position, where are the cells in the tissue context? Not only there are cells, but where are they? Are they inside the tumor? Are they outside the tumor? If they're outside the tumor, in the tumor microenvironment, where? Close to the tumor, distant to the tumor, or what's the distribution? Is there a gradient in this cell population? So to conclude my presentation, I hope with this short overview, I could show you that TissueFax platform can indeed contribute to a more robust experimental design, to providing standardized observer-independent data on the single cell level. There are a number of publications in which a comparison of our technology has been provided to, like, what is still called the gold standard visual evaluation. For example, here we have a counting of FOXP3 positive cells in this sample of colorectal cancer, which I showed earlier. 3 observers counted a number of regions manually. Remarkably, but not surprisingly, each observer got different counts. So every time, even if the same person counts twice, you get 2 different results. The median was formed of these 3 observers and then compared to the automated counting by our software, showing overall a very, very good correlation coefficient. So I hope with this introductory presentation, I could show you that with TissueFax, we can indeed support the research and diagnostic community to move from image to data and thereby increase the observer independence and reproducibility. I would like to point your attention to our web page. I mentioned at the beginning we have almost 1,800 reference papers published from our clients in more than 60 countries on all continents. Under here and on our web page, you will find this is updated every month. You will find always the complete list of known publications. It's currently 1,785 papers. You can sort or filter by certain topics or search for certain keywords like organs or diseases or molecules you are interested in, and you will immediately find who has already published what using instruments and software of TissueGnostics. To conclude my presentation, I would like to point also to a review article which my R&D group in TissueGnostics and our partners at the Queensland University of Technology have published earlier this year in the journal Genes. It's a next generation digital histopathology of the tumor microenvironment. So, which is showing some applications that we have done, but it's a review article which also shows an overview of the technologies available in the market today. I would like to thank our academic collaboration partners around the world, in Europe, also in Brazil, and in particular our partners in Australia. And finally, show you or invite you to submit an article. I'm a guest editor in Frontiers in Medicine, and I'm editing a research topic that is called Current Advances in Precision Microscopy. So if you or you have colleagues who have an article or considering to publish something, I would be happy to consider your manuscript. Please submit it. The call is open until April 2022, so until next year. Thank you very much for your attention. I hope I could show you that with TissueGnostics and our TissueFax platform, you can explore cells, the fundamental unit of life, at the precision that inspires. Thank you very much for your attention.